Friends, welcome to Biology Made Easy. We have discussed different interallelic gene interactions like incomplete dominance, codominance, and multiple alleles. Then also we have discussed that there are some non-allelic gene interactions or intergenic gene interactions like complementary genes, supplementary genes, epistasis, duplicate genes, polymeric genes, and collaborator genes. We'll discuss one by one. So today in this video, we'll discuss the complementary genes. Complementary genes, it gives a ratio of 9:7. So what are complementary genes? Usually the complementary genes are non-allelic. There are two independent pairs of genes present on different loci or even different chromosomes which interact to produce a new trait, but when they are present individually, they do not have any effect. That means, if you write the definition, two independent pair of genes, two independent pairs of genes present on different loci interact to produce a new trait, but individually they do not produce a character. So this the complementary gene action is best explained by Beteson and Punnett. Best explained by Beteson and Punnett in case of sweet pea. Sweet pea, Mendel worked on garden pea, Pisum sativum, but sweet pea is Lathyrus odoratus. Lathyrus odoratus. So in this Lathyrus odoratus, he found that there are some flowers which are white, while there are some flowers in some plants which are completely purple in color. So when he studied why such things are found, he found that for synthesis of the colored pigment, two independent pairs of genes are required. So how the whole thing is happening, let us see. In this plant, he took two white flowered plants. One has genotype small c, capital P, and the other has capital C, small p. This is also white, this is also white. Now when a cross was made between these two, then the F1 hybrid will be capital C, capital P, small p. These are the parents and this is the F1 hybrid. And he found that this F1 hybrid was purple in color. It was purple in color, while the two parents were white. So when again this was analyzed, again when this plant was self-pollinated in the F2 generation, what happened? In the F2 generation, the gametes will be of four types: capital C, capital P, capital C, small p, then small c, capital P, and small c, small p. So in this side also, capital C, capital P, capital C, small p, small c, capital P, and a small c, capital P. Now, if the checkerboard will be drawn, now see, this one will be capital C, capital P, capital P. This one will be capital C, and capital P, small p. This one capital C, small c, and capital P, capital P. This one capital C, small c, and capital P, small p. This one is capital C, and capital P, small p. Then this one, the capital C, and small p. This one capital C, small c, and capital P, small p. Then this one capital C, small c, and small p. This one capital C, small c, capital P, small p. And this one capital C, small c, and capital P, small p. This one small c, then capital P, small p. And this one small c, capital P, small p. And this one capital C, small c, capital P, small p. Then this one capital C, small c, and small p. And this one small c, capital P, small p. And this is all are in the lower case. So here you see, here both capital C, capital P are there. That means all these, they are both the dominants are present, capital C. So this will be purple color, this will be purple color, this will be purple color. That means where both capital C, capital P are there, that purple color. Similarly here capital C, capital P are there, that means they will be also all purple in color, where I have given tick marks. Then again you see, here capital C is there, here capital P is there, this one, and here this side capital C is there, this side capital P is there. So this 9, they are this 9, that means 9 by 16, these are purple in color. Now in the remaining cases you see, in the remaining cases, here only capital C is there. So this is white. Here only capital C is there. This is also white. And then here also you see only capital P is there. So white. Here one capital P is there. White. Here only capital C is there. White. Here only capital P. White. Here there is only lowercase, both C and P are there. So this is also white. So you see count white, 1, 2, 3, 4, 5, 6, 7. That means 7 by 16 are white. That means here it is coming in a 9:7 ratio, which is a modified ratio of 9:3:3:1 given by Mendel. Because in both the dominant genes are present in 9 cases, so it is giving purple color. While when one dominant is present, in this case, in the other case another dominant is present, and here no dominant gene, so these are clubbed together and it is 7. Now the question is, what is the reason behind it? Why such things are happening? Now the reason is, usually when a color is produced, the color is produced due to the anthocyanin pigment. And the anthocyanin pigment is produced through several biochemical reactions from the raw materials. So let us explain it. Suppose the raw material is A. Now this raw material will be converted to the intermediate product, which is B, and the intermediate product will be converted to C, and this C is anthocyanin pigment, which is purple in color. And the precursor of anthocyanin pigment is chromogen. And this is the raw material. So suppose for conversion of the raw material to intermediate chromogen. So this intermediate chromosome, it is colorless. Now, when the raw material will be converted to B, this is regulated by the genes. This is regulated by gene C, dominant gene C. And the dominant gene C, it produces one enzyme, and that enzyme controls the conversion of raw material to the intermediate product, that is chromogen, which is colorless. So this character is present in this case. So this character is present in small c, capital P. You see, for the conversion of raw material to chromogen, the intermediate product, it requires the function of the dominant gene C, which will produce enzyme, and that will convert the raw material to chromogen. But in case of plant, small c and capital P, there is no dominant gene C. That means it cannot convert the raw material to chromogen. That means the flowers will be white in color. Similarly, in case of the other plant, when the chromogen will be converted to C, it requires the role of another gene, that is here the capital gene P. So the capital gene P, it produces one enzyme, and that converts the intermediate product chromosome to anthocyanin, which is purple in color. And this is usually present in the plant, capital C, small p. So in this plant, capital C is there, which can convert raw material to chromogen. But chromogen, when will be converted to anthocyanin, it requires capital P, and that is not present in this plant. So when in this plant only this type of genotype is present, it cannot produce anthocyanin pigment, so it remains white. So now it is clear that in the plant small c, capital P, the flowers will be white, because for the conversion of this raw material to chromogen, that dominant gene C is not there. Similarly, in the plant capital C, small p, the flowers will be white, because for the conversion of a chromogen to anthocyanin, the capital P, dominant gene P is required. That is also not present. So naturally the flowers are white. But when both are present together, in case of F1 hybrid, what is happening you see? In that case, when both are present together and both capital C and capital P are present, as in case of the hybrid, what is happening? Now the capital C, which is present in this white plant, that is converting the raw material into the intermediate chromogen, and the other plant where capital C is there, as in this case, that is converting the intermediate product chromogen to anthocyanin, which is purple in color. That means for production of anthocyanin or purple color, it requires two independent pairs of genes. Here one is C, the other is P. So here the gene interaction between capital C and capital P is complementary to each other. That is why they are known as complementary gene interaction. Now further it can be proved when the flower extract of this white plant was taken individually, it was white in color. When the flower extract of this plant was taken, it was white in color. But when the flower extract of the two plants are mixed together, immediately converted to purple. Why? Because both the genes are present together. So from both these cases, from the flower extract as well as from the crush, it is clear that the action of genes C and P are complementary to each other. This is known as complementary gene interaction. And it is giving a modified ratio of 9:7 instead of 9:3:3:1. So this is about complementary gene action, and the example is Lathyrus odoratus. I hope now you have a clear understanding of the what is complementary gene interaction, what is the biochemical pathway. Thank you very much. God bless you. Have a good day.